Good afternoon. Welcome back to Grand Rounds today. Please remember to sign the attendance record at the back of the auditorium, and also please remember to fill out and turn back in the program evaluations. And please, if you could, give us any ideas in regards to future topics or future speakers that you'd be interested in learning about. Today, it's my pleasure to introduce Dr. Adam Dupuy. Dr. Dupuy attended undergraduate at Central College, and then he moved up to the Twin Cities, where he received his PhD in molecular, cellular, developmental biology, and genetics. Then moved back to the University of Iowa, where he is now associate professor at the Carver College of Medicine at the university in the anatomy and cellular biology department. He's been extensively published in the area of cancer genomics, and we're pleased that he was able to accept our invitation today to update us on current concepts and trends in cancer genetics. And please join me in welcoming Dr. Adam Dupuy.
Thanks. All right. Can everyone hear me? All right. Okay, great. So thanks for that introduction and the invitation to be here today. I just want to run through a few concepts here once this gets going. Okay. So this is the structure of the talk I'm going to be giving today. So first I wanted to review, I wasn't sure how many of you are involved clinically with cancer care, so I wanted to review for those of you who are not, just sort of basic concepts in cancer genetics. I'll describe the technology of current genome sequencing methods that are used, both for research and clinical applications, how we use that information then to interpret the genomes of cancers, which are quite complex, and then a couple examples of how this information's been applied to the care of cancer patients and where things are going. So as some of you probably know, there are some inherited cancer predisposition syndromes. The most common are, say, BRCA1, BRCA2. This is inherited breast and ovarian cancer. P53 mutations would be Li-Fra-Mani, inherited colorectal cancer, APC. There are any number of inherited cancer predisposition syndromes. These are rare, though. All summed together, they account for only about 10% of total cancer incidence in the country. Instead, most tumors arise sporadically in families, so not through inherited predisposition syndromes, but occur just sporadically due to the acquisition of somatically acquired mutations. And so, let's get rid of that. So the one thing we do know is that mutations occur throughout the cells of our body as we age. And there's really, unfortunately, so far as we can tell, not much we can do to avoid this. There are about 50,000 DNA damaging events in each of your cells every day. That can be increased through exposure to carcinogens and radiation and any other number of things that can cause DNA damage. Fortunately for us, there are just about right around 200 proteins encoded by our genomes that are involved with repairing these DNA damaging events. Nevertheless, as we age, these mutations, some of them fail to be repaired, end up permanent fixtures in our cells, and depending on how those mutations affect the behavior of cells can lead to sort of outgrowth or clonal expansion of cells within the tissues of our bodies. This is a paper that was published just last year, and what this group showed by analyzing the genetics of tissue taken from the upper eyelids of many patients, what they did was cut the excised skin into small pieces and analyze the genetics of just small little regions of the eyelid, and found that as patients age, they could find cancer-associated mutations in normal-looking eyelid skin. But nevertheless, these sort of clonal expansions of these populations of cells are there. They harbor these mutations that are associated with cancer, but the cells had not yet developed fully into a tumor. So this is a recent example of what we've suspected for many years. So what can we do, really, to reduce risk of cancer? Well, there are some things we can do. We can limit environmental exposures and make decisions that will reduce DNA damage. These would be dietary changes for those of you who have teenagers, avoiding tanning beds and the like, things like this to reduce risk of skin cancer. All of these things can reduce DNA damage and reduce risk of getting these mutations fixed in our cells. But occasionally we can have incomplete DNA repair, and those things can be increased if you have genetic defects in some of the proteins involved in DNA repair. As those things accumulate in our cells, they're thought to contribute to a number of different disease states, including cancer, some immune disorders, accelerated aging, and maybe even some neurological disorders. So we're constantly facing this sort of balance between DNA repair and mutation. And depending on which way the scale tips, we will reduce risk for these disorders by increasing DNA repair mechanisms and decreasing the amount of DNA damage. And if we go the other direction and have a situation where our cells accumulate mutations at a higher rate, then likely it will increase risk for these disorders. And the reason that is because cancer really is the net result of an accumulation of mutation over many years, decades most likely. So as cells acquire mutations here, the little lightning bolt is meant to be a mutation or a DNA damaging event, it can change the behavior of a cell. A pathologist may not be able to look at this cell and notice a difference at the microscopic level, but within the cell, the cell will begin to be rewired and its behavior will be changed by this mutation. That can lead the cell to grow at a faster rate or survive and begin to dominate in a little clone within a tissue of the body. And over time, this cell or some of its daughter cells will acquire even more mutations. And given enough time and mutations, a full-blown tumor can emerge. This is sort of the process as we've understood it for many years. There are these nagging questions, though, that we're beginning to be able to address with genome sequencing, such as how many mutations are found in an average tumor. We know it requires many mutations, but how many exactly? Which gene mutations cause cancer? There are over 20,000 genes in the human genome. Which of those are associated with cancer when mutated? And then, most importantly, can we do anything with this information? Can we use this information to improve cancer therapy and improve outcome for patients? So these are some of the questions we'll delve into here today. And I'll start just with a little more review of basic cancer biology here. What this is, and you'll notice as I go through this talk, I'm going to hop around a bunch of different tumor types. I'm a cancer geneticist. I'm interested in cancer genomes. I don't really care what type of cancer they are. We, in my lab, do research on about a half dozen different types of cancer. So I'll use many examples from different types of cancer in the talk today. This one comes from colorectal cancer. You can see here on the top left a normal cross-section of normal colon tissue. You can see the colon crypts here. And if you were to cut across this in cross-section, they'd appear this way. And what the arrow is indicating here is an early lesion that is heading toward the direction of cancer. It's adenomatosis crypt here. You can see its intense staining. This can go in two different paths. It can become a tubular adenoma or a villous adenoma. And we know that there are mutations associated with these various transitions, these pathological transitions. The tubular adenoma can even become more, this tissue can become more elaborate. Blood will pool here in the stalk of this tubular adenoma. There are mutations that are associated with these sorts of transitions. This yet is still just an adenoma, not a full-blown cancer. That would be an adenocarcinoma. These are the things that can be removed with colonoscopy and will dramatically reduce risk of colorectal cancer. But if not caught early, they can progress to carcinoma and metastasize to other sites in the body, such as the liver in this instance here. So this is a multi-step process to go from a normal-looking tissue to this early lesion to these benign tumors all the way to malignant metastatic carcinoma. And there are mutations involved in making these transitions at each of these steps. And in some cases, we have a reasonable idea of what these mutations are. We do have at least a reasonable idea of the timing of these events for a number of different cancers. The average age of onset for many types of cancers is quite variable, but for example, in men, prostate cancer generally is quite late in life, whereas other types of cancer can crop up earlier. Leukemias in kids can be quite common. Acute lymphoblastic leukemia is the most common form of cancer in children. And that is attributed just to the biology, the varying mechanisms involved in these types of cancer. And in some cases, we know the sort of risk factors, smoking, for example, associated with lung cancer, and we can mark the transition in the tissues as we get normal-looking tissue to something that looks more like cancer in situ here, and then a full-blown malignancy. So again, cancer is a multi-step process, and the genetics for some of these types of cancer have been worked out fairly well. In other cases, not as well. So as I go on here, I'm going to talk generically about cancer driver mutations, and I just wanted to touch on what that means. A driver mutation in cancer is a mutation in one of the genes in the genome that causes the behavior of the gene. And generally, cancer drivers come in two flavors. There are tumor suppressor genes, and then there are oncogenes. Tumor suppressors, as the name implies, are genes whose function it is to suppress growth, to maintain the integrity of the genome, behaviors like this that would suppress or reduce mutational burden and restrain cell cycle. Oncogene, on the other hand, is a gene that encodes a protein that contributes positively to cell cycle, something that promotes proliferation, something that drives cell invasion or other sorts of behaviors that might promote cancer. Now, you might ask yourself, why does our genome encode oncogenes if they can produce tumors? Well, we all need to grow from a single cell to an adult organism, and that requires a tremendous amount of cell proliferation. We need to maintain our tissues, which requires a lot of cell proliferation. So we would not be able to function or maintain tissue homeostasis without the function of these oncogenes. So it's, again, a fine line that we have to walk, our cells have to walk between proper regulation of these things. So that's just a brief overview and some cancer biology and cancer genetics concepts. And now I want to talk briefly about some of the sequencing methods that we're using currently to sequence cancer genomes, and really any genomes. This is an area of technology development that is moving quite rapidly. As I'll show you, there's been a lot of technology improvements over the course of the last decade. And so I'll just briefly touch upon some of the strategies that are currently being used to do genome sequencing. There are a number of companies using an approach called emulsion PCR, where this PCR stands for polymerase chain reaction. It's essentially DNA synthesis in a tube. In this case, little beads are used and isolated in little droplets of oil, and DNA synthesis is done on the surface of the bead. These beads are then subjected to DNA sequencing. So isolate millions of these individual beads, each with a specific strand of DNA, and you sequence enough of them, and you can, in fact, sequence a genome this way. And there are a number of companies that use this process, 100 to 200 million beads to get this process done. The more common, predominant technology right now is solid phase amplification. This is the method that we use in my research more often. Here, DNA synthesis is done on the surface of a flow cell or of a little microtiter chip. And here we can build 300 to 400 million little DNA clusters and sequence them on the surface of this chip. And this is done using nucleotides that are fluorescently labeled, and so you can actually image the DNA synthesis happening and pretty much in real time and know the sequence of the DNA that's being synthesized and get genome sequence this way. The combination of these technologies that have been introduced over the past 15 years or so have dramatically reduced the cost of DNA sequencing. So when I defended my thesis and got my PhD in 2001, the cost of sequencing a genome, the first draft of the human genome was published. At that point, to repeat the process would have run us about $100 million. So that's quite expensive. But since that time, we've gone from $100 million down to now about $1,000. So this is a price point at which we're approaching probably common genome sequencing in clinical practice, where laboratory tests are not too far from this range. So now that's just to get the raw sequence. The difficult part is interpretation, as I'll show you in a second. But we're getting very close to clinical sequencing being a reality here in the last couple of years. So I think we're going to be in for some changes soon. When that occurs will determine largely by when insurance companies decide to cover the cost of genome sequencing on a regular basis, probably soon. So now that we can sequence genomes, and at the University of Iowa, at our cancer center, we're gearing up to do this hopefully in the near future for every cancer patient that comes to our clinic. We want to figure out how to interpret that information. We can get the information now. It's not that expensive, but how do we interpret it? How do we understand it? And in part, this has been aided by this entity that the National Cancer Institute has funded partially called the Cancer Genome Atlas. It's a NIH, NCI-funded effort that's been going on for the past 7 years or so. The goal of this effort was to amass a huge collection of tumors from all over the country and a few other sites outside of the U.S., sequence the genomes of these tumors along with the patient's normal DNA, and catalog all of the mutations that are found, and also collect pertinent clinical information and see if we couldn't make correlations between specific mutations and clinical outcomes in these cancer patients. The net result is a tremendous amount of information, 2.5 petabytes. If you're not familiar with what a petabyte is, that you'd have to write that out on about 200,000 DVDs, so it's a lot of information. We're currently at sort of terabytes. Maybe some of you have 1 or 2 terabyte hard drives on your computers. Petabyte would be the next step in this process, so it would be 1,000 terabytes. It's a lot of information on 33 different tumor types, 10 of these being very rare cancers. So far, 11,000 patients have participated, and there are 7 different types of data that have been collected on every single one of these patients, every single one of these tumors. And the information has been quite useful. And in one case, in the case of stomach cancer, analysis of the data led to the discovery of stomach cancer actually being 4 distinct clinical entities. It was not appreciated before. That could only be revealed from the clinical sequencing of those tumors. So there's a lot of information here. We're still continuing to mine this information, which is part of what we do also in my laboratory. But this has allowed us to gain some insights and answer some of the questions that were raised earlier here when I spoke about the evolution of cancer. So how many mutations do we have in an average tumor, and which gene mutations cause cancer? We can now begin to get answers to these questions. And so when we talk about which genes are driver, how many cancer drivers are there, we now start to talk about mutations in tumors in terms of these general labels, drivers and passengers. And we use these terms because they denote sort of our thoughts about the effects of the mutations. A driver mutation is the mutation that actually contributes to the malignancy, that's somehow responsible for transforming the cell or maintaining the tumor or affecting the behavior of the tumor clinically. Passenger mutations are simply background. So they're in the same car, they're in the same vehicle, they're just along for the ride, but they're not really contributing anything meaningful to the tumor. And so from a research perspective, we want to focus our emphasis on, or in our time, on understanding the biology of driver mutations and not get distracted by inadvertently studying passenger mutations. Now, if we're going to have an easier job of this, we would hope that driver mutations, the ratio of driver to passenger mutations would be favorable, something more like a little Mini Cooper, right? 1 driver to maybe 2 or 3 passengers. It's also possible that the situation could be much worse. We could have a single driver in a bus full of passenger mutations. And so the Cancer Genome Atlas allowed us to finally get a glimpse into this question, and unfortunately it was bad news. It's actually worse than the bus scenario even. The mutation rate in some cancers is tremendously high. Melanoma, for example, has about 10 mutations per megabase of DNA, which is actually a lot. There have been sequenced melanomas that have 1 million mutations. The vast majority of those are going to be passengers. But with such a large background signal, with so many passenger mutations, it's hard for us to identify the drivers within them. So this is an ongoing challenge that we face in the field of cancer genetics. And the situation is a little more complex because the pattern of mutations and the frequency of mutations is extremely variable across tumor types, as you can see here with this chart. So as you increase on the y-axis from the bottom to the top, the mutation number increases. And you can see various types of cancer here, and individual tumors within each type of cancer that have been sequenced. And the distribution of mutation frequencies is quite high, and the characteristics of the mutations are also different. So down in the bottom, you don't need to really understand too much what's going on here, but just notice that the color distribution for each type of cancer is a little bit different. And that means the type of mutations that are observed or the mechanisms of mutations are also variable across tumors. And so this makes it quite challenging to understand what's going on. So how then do we decide what is a driver mutation and what's a passenger mutation? Well, a lot of it has to do with this figuring out the mutation frequency within a tumor cohort, and then looking for or correcting for gene size. Genes are a variable size. You would imagine a massive gene like one of the larger genes in our genome is called Titin, appropriately. It's a massive gene. You would expect randomly mutations to land within Titin much more often than a small gene, and so we need to account for that. We also need to account for the nature of the gene mutations, as I just indicated. The type of mutagenesis that drives various types of cancer is variable, and so we want to model that as best we can. And so if we build, right now, we're building complex computational models that require quite a lot of computer time to run to interpret or identify driver versus passenger mutations within individual tumors. And you can see here some of the results of that analysis. When you look at these various types of mutations that can be seen within tumors, you can see that in this case the tumors are color-coded, the little circles are color-coded. You can see very distinct stripes of tumors that fall within these sectors on this graph. So a mutation is not just a garden variety mutation. The type of mutations that drive cancers, the properties of them are quite distinct for various types of cancer. And this was somewhat of a curiosity as well when this was discovered. Sometimes it's not surprising. For example, melanoma here in black. This type of mutation is a type of mutation that's caused by UV exposure, and so this was not surprising, right? Melanoma is driven largely from UV damage. But some of these other forms of cancer, it's a bit of a mystery in some cases why the particular patterns of mutations occur here. There's probably something going on, and this is telling us something about the biology of these tumors, but we don't quite yet understand what that is. So research in this area is still ongoing. In terms of which genes in our genomes are drivers, there are over 200 cancer genes, so tumor suppressors or oncogenes, that have been identified through genome sequencing so far. And this is just a distribution of these genes. Anything in the second quadrant up here, these are driver mutations that are seen often in many types of cancer. And you can see that there are a lot of genes here. Some of them are well known, some of them have just recently been identified. In some cases, we don't even really yet understand the biology of the protein encoded by this gene, so we don't even know what the mutation actually is doing in terms of cell biology or gene expression or what have you. So there's a lot of research to be done here to understand the nature of these mutations specifically. Some genes, like quadrant 3, these are genes that are mutated at a high rate in only one tumor type. So for reasons, in some cases we understand, and others we do not. It's specifically associated with a particular form of cancer, not broadly seen in many forms of cancer like these genes, or these here in quadrant 1. So in all cases here, 200 genes, in order to do something about or use this information to improve clinical outcomes for patients, we need to understand the nature of the mutations that occur in these genes, understand how the mutations affects the gene function and protein function encoded by that gene, and then determine, is there something we can do to target that mutant protein, or in cases where the protein is broken permanently, replace its function if possible. And so it's a tremendous amount of research that is required to sort all of this out, and will take quite some time. But I want to tell you about just a couple of examples of how genome sequencing has been used clinically. And so these are a couple of complex examples. One here is a story seen in melanoma. It's a for me, melanoma is sort of a bright spot in cancer research. When I was a graduate student, patients diagnosed with metastatic melanoma, 5-year survival was quite low in the single-digit percentages. There was nothing you could really do. Standard chemo was given to these patients, but generally the tumors did not respond. So it was not great at that point in time. But since then, we've had a lot of developments, and things are looking quite favorable now in melanoma. And one of those discoveries was that this gene in our genome called BRAF is frequently mutated in melanoma. So 50 to 60% of patients have a recurrent mutation. So it changes this amino acid to this amino acid at position 600. When you do that in the gene BRAF, it turns it on permanently. Normally this would be a kinase that is regulated, its activity is regulated. But when mutated like this, BRAF is constantly on. And when that happens in melanocytes, it causes them to grow out. And in fact, if you have moles, like I do, a good number of them actually have this mutation in them. A frightening thought, but it is a very early initiating event in melanoma and is associated initially with benign outgrowth of these just melanocytic nevi in the skin. But it just starts the ball rolling. Now, every single tumor cell for patients who have this probably have this mutation. A drug company nicely made a drug called vemurafenib, which inhibits specifically the mutant form of BRAF, does not inhibit the wild type. That's important because this is an important protein in many of our cells, and inhibiting its activity systemically would probably be quite toxic. This drug specifically targets the mutant form. It's an orally active inhibitor of this mutant form of BRAF, and in an early clinical trial had rather remarkable, outstanding results. So here is a patient, 38-year-old male with a BRAF positive melanoma. This is not a typical presentation for advanced disease, but this individual had all sorts of subcutaneous lesions, melanoma throughout the skin, so it's a very visual example of how remarkable the drug is. So after 15 weeks on vemurafenib, this individual was almost fully recovered. The other advantage of these targeted therapies is the toxicity profile is quite low. So about the worst thing that will happen to you with vemurafenib is maybe a little bit of a skin rash. So this is about as close to a magic bullet as we have gotten so far with cancer. The unfortunate news is that the effects of this drug are limited in their effect. So 15 weeks of drug, patient looks great. 23 weeks later, though, the patient has relapsed on drug. And this individual did not survive. And the story of what's going on here is very similar to if you understand the genetics of antibiotic resistance in bacteria. It's a very similar story here. The drug leads to the death of the vast majority of tumor cells in the patient, but a very small population of cells persist that are resistant to this drug. And when the patient recurs and the tumor grows back, the tumor is now entirely resistant to the drug, and the drug's no longer effective. We know in a small percentage of patients that this is driven by mutations in some known proteins, and so combination therapies are being tried now. We think that the resistance is in fact determined by other mutations within the tumor, and one of the projects within my lab right now is to understand the genetics of vemurafenib resistance so that we can improve the duration of this therapy, making it more effective, hopefully long term. So that's one example of how genome sequencing has led to an improved therapy. First, the identification of BRAF, development of a drug targeting that mutation, and now understanding the acquired resistance of vemurafenib response in cancer cells. So that's all work that's ongoing. And one aspect of that drug resistance suggests that tumors, when they develop, are genetically heterogeneous, meaning they're not made up of the same tumor genomes. It's sort of a hodgepodge of different tumor variants. And this paper that was published in the New England Journal back in 2012 really drove this point home. So in this paper, a group was looking at patients with renal cell carcinoma and isolated both the primary tumor, in this case here on the left, the kidney, as well as other metastatic lesions throughout the bodies of these patients. So I have to say, every time I talk about this paper, it's important to point out these patients really did not need to undergo this surgery at all. They consented and did it just for this study. There was no therapeutic reason to go in and resect these metastatic lesions. They did it solely for research purposes, which was, I thought, quite impressive for these patients to volunteer to do. But the information gained from this paper has been helped quite a lot in how we think about cancer. So it's, I don't know, important to acknowledge, I think. So what this group did here for multi-region sequencing in these tumors, they took the primary tumor, quite large, 10-centimeter chunk here, and just cut out little regions of it in various locations throughout the tumor mass, and sequenced each one separately. They also sequenced different metastatic tumors throughout the body of this patient. And what they found is that there are, not surprisingly, lots of mutations. So across the top here are the gene names that were found to be mutated. If the box is gray, so here are the individual cores here on the side. Here are the primary tumors, pre-treatment and post-treatment, and on along up here to the metastatic tumors at the top. And what you can see, if it's a gray box, it means that gene is mutated in that sample. So there are a lot of mutations that are common to all tumor samples taken from this one patient. Not surprisingly, because cancer is a clonal disease. It emerges from the same cell initially. But then as the tumor begins to spread, you can see that now there are unique mutations that are only found in the metastatic tumor, that are not seen in the primary tumor. And then there are also some primary mutations in the primary tumor that are not seen in the metastatic tumor. So really, what this tells us is that this patient does not have one tumor, but actually a collection of many related tumors. And you can see that here. They can piece together the timing of these events, actually. So the patient started out with normal tissue, suffered a mutation, in this case, in a gene called VHL. That got the tumor going. Once the tumor spread, though, it began to accumulate mutations in the primary tumor and the metastatic tumors differently, because these tumor masses are geographically separated in the patient's body. And so they begin to drift genetically away from each other. What this means, though, and this was seen in a second patient here, actually, similar trend. You see this sort of genetic drift in the primary versus the metastatic tumors. And in this patient, the first patient that I described to you here, it was also appreciated that there are mutations in a gene called mTOR. This protein can be targeted with an FDA-approved inhibitor, in fact. And if you had sequenced the right spot of this tumor, you'd say, oh, look, there's a mutation in that gene. We should give the patient this drug. But what you would realize is that what you wouldn't realize at the time is not all of the tumor would be responsive. So again, you'd get a similar result with this patient as that melanoma patient. Some of the tumor would respond, some would not, and eventually the patient would relapse with resistant disease. So it is quite a complex situation that's happening within patients with metastatic disease. And not only do we need to sequence the patient's tumor, we probably need to begin thinking about sequencing multiple different biopsies from patients with metastatic disease and trying to understand these patterns of evolution within the tumor and try and find mutations that are not out here that make these masses distinct, but target mutations that are shared between all of the tumors throughout the entire patient's body and hopefully have better outcomes there. So this is information that I think we will eventually need. We are not quite ready to do this on a regular basis, and in some cases it may be very difficult to get such biopsies from patients, but so there's a lot of developments that need to occur before this can become commonplace. So at the end of the day, what this simple depiction of tumor evolution is actually much more complicated. So a primary tumor gets started. Tumors begin to shed cells into circulation or through the blood or lymphatic system. And as those tumor cells lodge at distant sites in a patient's body and begin to grow out as metastatic lesions, they'll begin to drift genetically away from that primary tumor mass, and setting up a situation where you could have variable response to therapies within an individual patient. So we're not really talking about a single tumor genome here, we're talking about many tumor genomes. And so it's quite a complex situation and more research certainly needs to happen to understand this process and how it happens in each individual patient. So what do we know right now from the current genome sequencing that's been done? So we know that each patient's tumor is unique and complex. We know that cancer genomes do not share many mutations. So that example of the BRAF mutation found in 50 to 60% of melanomas actually is an exception. It's not the rule. Most driver mutations are found in 3 to 5% of tumors of a particular type, so they're rare. They're certainly associated or have been verified to be driver mutations, but they're not common. So this complexity between patients and even now within patients sets up a situation where it's very difficult for us to understand the key genetic events that drive cancer development and invasion and metastasis. So we need better experimental models to test these mutations, to understand these trends, and to develop better drugs that are more effective at treating these mutations that occur earlier in the process. So these are the questions really, at least in my mind, what we need to know now. And in some cases, we're working on some of these things in my laboratory. So we want to understand how much of the genetic complexity is meaningful. So as I described earlier, defining a gene as a driver mutation is in large part due to a frequency calculation. Is it mutated at a rate higher than we would expect to see by chance? That's completely a function of how many tumors we've sequenced to date, and that's somewhat cash restricted, because while $1,000 genome is much cheaper than the $100 million genome, it's still more money than most research labs can cough up for an individual project. So we need to understand better how much of this complexity is meaningful, how much of it is noise, how much of it is signal. We want to know if we can effectively target a large number of mutated genes with specific drugs. There are already a few dozen FDA-approved drugs like vemurafenib that target specific mutated gene products in tumors. We need to do a better job in understanding or identifying driver mutations so that our colleagues in drug development can make drugs targeted to these driver mutations or the mutated protein products. And are there other therapeutic approaches that could target cancer more broadly? Maybe not targeting specific mutations, but taking a step back and looking perhaps more broadly at cancer. And so I wanted to finish with this one slide because it is, again, for melanoma and other forms of cancer, sort of a surprising and really exciting development that's occurred over the past few years. This type of therapy sort of exists at the border between immunology and cancer genetics. So as hopefully you would understand now, our cells of our body begin to drift genetically through the acquisition of mutations, and tumors have lots of mutations. And it turns out that there's one cell type or a handful of cell types in your body that are meant to identify foreign cells within your body, right? And that's your immune system. And one problem we thought naively with cancer and why it's so difficult to treat is that it's really derived from your own cells. And so your immune system, we thought, couldn't recognize it because it's really you. It turns out that's not entirely true. As tumors develop, they acquire so many mutations that they begin to look not quite like self, so not like the self that your immune system was familiar with, and you can begin to see immune responses to tumor cells. But the tumor cells, through sort of this constant evolutionary process, evolve mechanisms to suppress that immune response. And I've identified some of the mechanisms involved with that. And there are now new drugs, a new class of inhibitor drugs, these checkpoint inhibitors, that prevent the tumors now from suppressing the immune response. And in some patients, we're convincing the patient's own immune system to attack the tumor. And in some cases, we're getting durable responses. So patients that are still tumor-free 10 years after being treated. The other advantage of this immune therapy is that the immune system can go anywhere in the body and attack the cancer anywhere where it exists. So we're seeing some remarkable outcomes in patients with metastatic disease. Now, you would say this is cancer immunotherapy, not cancer genetics, but one thing we've learned is that the higher the mutation burden of the cancer cell, the more likely this therapy is to work. So the more likely the immune system will be able to see a tumor as being foreign. If it has 1 million mutations, the immune system's more likely to recognize it as something not quite self. So this is a way in which we can sort of merge my field, cancer genetics, with immunology and still try and improve cancer therapy with a broader approach, not just a targeted therapy, but just basically revving up the immune system to attack a patient's tumor. So we're getting remarkable results here. So we have a lot of irons in the fire right now, and we're making these discoveries at a pretty surprising rate. So I think with that, we've got a few minutes left. I'd be happy to answer any questions you might have. Thanks. Yes.
Kind of in the same vein as the immunology approach, I'm in neurology, and our life is spent looking at cells deteriorating and dying and going through apoptotic decay. Is there any interest in trying to enhance or finding a way why the cancer cells don't activate an apoptotic pathway and just die on their own? I mean, they must shut off that whole process, right?
They do. So another class of driver mutations are genes that regulate apoptotic pathway and responses, and there are a number of them that are known that are mutated often, sort of as a collection of genes. They are mutated often in tumors, and so they no longer respond and undergo apoptosis appropriately. There are drugs, actually, that are in clinical trials, and maybe a couple that are FDA approved that do target some of those mechanisms, yeah. Now, applying them to other areas of clinical practice, might be beneficial. That could be. Any other questions?
Is there a difference in the cancer genome from a solid tumor that doesn't metastasize and just remains a mass in the body versus one that's capable of metastasizing?
Yeah, that's a great question. At a genetic level, is there a difference?
Yes.
Yeah, so to my knowledge, not that we can tell yet. And some of those trends seem to be more a function of the cell of origin for the various types of cancer. So you would have a tumor like glioblastoma, which metastasizes very late, versus cancer like melanoma that can metastasize before a patient even knows they have melanoma. It could be a function of the biology of those cells. Melanocytes are migratory cells to begin with. Glial cells are not. So it could be controlled more from the biology, the cell of origin, than the genetics of the individual tumors. But the jury's still out on that one. Any other questions? All right, thanks.